Okay, good. So we'll start with Ruthania. Ruthania, what did we cover last week? Mod 6.
We covered substitutional mutations.
Good.
And
we looked at how to read this mRNA to amino acid table/chart. And you would see multiple different exam questions based off this. You can see if I go up here, they show you the same mRNA to amino acid chart, but as a sphere, versus here, they show it to you as a table. It's a very high yield part of Mod 6, right? And the key learning point is, when you go to DNA to mRNA, Matthew, what should you do to the sequence? If you're given a DNA sequence, you have to go to mRNA. What should you do?
Work out the complementary
Good.
base pairs and then change T to uracil.
Perfect, right? Those are two things you want to do. Complementary is the most important, and every thymine does not exist. Because remember, uracil is a more stable base. Remember, the goal of mRNA is to leave the nucleus, go into the treacherous cytoplasm where there's enzymes that break things down. Hence why T is replaced with uracil. It's to allow the mRNA to persist in the cell longer without being broken down by the cytoplasmic enzymes. Good, that's very important. Now, next point here, we talked about somatic and germline mutations. I'm going to ask a few of you some questions about this, right? So first of all, I'm sure we can all see here somatic mutations, somatic cells, body cells, right? Germline mutations are referring to sex cells or gonadal cells. What's the difference in chromosome number, Sarah, between a somatic and germline cell?
So a somatic cell has 46 and a germline cell has 23.
Good, right? Going to Kriti. Kriti, are all somatic cells identical and are all germline cells identical?
No.
So which one? So somatic cells, are they all identical? Yes or no? Okay, I think Kriti said no. Alyssa, what do you think? Are all somatic cells identical, Alyssa? And are all germline cells identical?
I think that all somatic cells are identical, but germline cells are not.
Very good, right? That's the answer you want to hear, because somatic cells are made by mitosis. You are copy pasting DNA. It's that simple, right? The DNA does not change. The only time it does change is mutation, right? And that's random. And that can either be spontaneous as an accident during DNA replication in mitosis, or it can be caused by external factors that we already spoke about, right? Radiation, chemical mutagens, biological mutagens. That's what the syllabus wants you to know. Germline cells are all different because of meiosis. Very good. So now let's talk about somatic mutations and germline mutations quickly. Sarah, actually I've already asked you, we'll go to Trissy. Are somatic mutations passed on? And are germline mutations passed on?
Somatic mutation is not passed on because it's not in the gametes, but then for germline mutation it is, because through gametes, when the fertilization take place, it will give it to the offspring.
Perfect, right? When you form an offspring, it's the gamete cells of the organism that are combining. So if you have a germline mutation, aka the mutation is present in your gametes, it is of course going to get passed on when those two gametes form to form a zygote. Very good. The one caveat I'll make, everyone, is although the syllabus emphasizes a somatic mutation is a mutation in somatic cells, a germline mutation is a mutation in germ cells. I want you all to understand this. For an organism to have a germline mutation, it is most likely also present in their somatic cells. Let me explain, right? So what needs to happen is there needs to be a healthy organism, number one, here, who gets some kind of mutation in their gonads, right? It could be from radiation to the gonads from a CT scan they got done 30 years ago. It could be from being in the sun. It could be from sitting in a spa too long, which can impact the sex cells, cause mutation as well, right? Some kind of reason has caused a mutation in their gametes, right? But they are still healthy because their body cells are fine. Now, when that gamete combines with an ova, the resulting offspring organism will have mutation in all of their cells because the spermatocytes that combined to form the zygote had the mutation. And so the zygote is going to have the mutation. Remember, every single cell in your body, brain cells, eye cells, hair cells, all originate from that zygote. So now every single cell in this offspring is going to have the mutation. This is still a germline mutation, but can everyone appreciate the mutation is also in their somatic cells? It is also in their germ cells. So exam questions will try to trick you, but just know most of the time, except for that first mutation that hits the gametics, the gonads of the first organism, all offspring will have the mutation in all of their cells. The only other caveat as well is, remember, mutations are passed on, or diseases that are genetic or heredity are passed on via patterns of inheritance. So if this was an autosomal recessive disease, this mutation could be masked if it's a recessive allele. If there is a dominant allele passed on from the other gamete, it's going to mask the mutation, and in fact, this organism will be completely unaffected as well. They'll carry the mutation in their cells, right? But they will be unaffected. So as long as you all understand, germline mutations typically affect all cells of the organism, not just the gonads, except for that first organism to unluckily be hit with the mutation in the gonads, okay? And the mutation is passed on, but whether they express disease or not is dependent on what you all learned in modified patterns of inheritance. Very good. What else did we learn last lesson? We learned quite a bit. Matthew, what is this stuff went about? And tell us, Matthew, is non-coding DNA important?
Yes, non-coding DNA is important, such as there was telomeres, gene regulatory roles. I forgot the other ones, but it is very important. It was coming to understand how important it is with the telomeres, and our idea every time it replicates, it slowly chips away at the telomeres until it no longer serves that purpose.
Good, yeah, good. I like your answer, and you gave two very good examples, right? The key thing that the syllabus wants you to explain, though, is that it's significant because it still can change phenotype. Even though non-coding DNA, remember it's defined non-coding because it doesn't actually transcribe and translate to protein, it will still affect phenotype. That is the word you need to have in your answer. So if you guys didn't think of that, write it down below here, right? When it's assessing the significance, you need to link it to phenotype being changed. And Matthew's completely right. So just very quickly going across, Alyssa, can you tell us a bit about gene regulatory sequences and how they can actually affect phenotype?
Basically, I think it's like they bind to they like.
I'll help you out with an image because I understand it's hard to appreciate, especially when you learned it 1 week ago and it's completely flipped your understanding of how genes affect phenotype. So let's give it a go.
Basically the proteins will like bind to the gene, I guess. I just remember that like it like does something to the transcription and translation process.
Good. Okay, so you've got the key concept here, which is these gene regulatory sequences are affecting transcription and translation of other genes. Now the question is how, right? Now, to really understand this, this is your gene regulatory sequence. GRS, or gene regulatory sequence, right? Maybe I'll just take any acronyms out because acronyms confuse me as well, right? So what I've just shown you there on the left is your gene regulatory sequence. You've got 3 genes that are downstream or sitting next to that gene regulatory sequence, okay? Now, the way this gene regulatory sequence works is it allows other chemicals or hormones to bind to it. And when those chemicals or hormones bind to it, it'll send signals downstream that affect the amount of transcription or translation of gene 1, gene 2, or gene 3. So I'll give you a perfect example, right? Let's say in another chromosome, there is a gene. Now, I'm sure you all know genes make proteins. One of the functions of proteins are hormones, chemical messengers. So this gene could transcribe and translate and produce the hormone testosterone. Now, I'm sure you all know, use your lay people understanding. Testosterone, you might know gym people sometimes use it to get naturally or artificially, increase the muscle mass. And it has a lot of health effects, but in this note, this is exactly how testosterone works. Testosterone is made by a gene, right? And it will go and bind to a gene regulatory sequence. And that will tell the gene regulatory sequence to send signals downstream that'll increase the transcription and translation of genes that make muscle tissue. Now, this gene 1 could be the gene that makes the muscle protein actin. This gene 2 could be the muscle protein that makes myosin. Gene 3 could be tissue that makes, or a gene that makes the protein collagen. And so now you understand testosterone and exactly how it acts. It binds to gene regulatory sequences that will go and increase the production of various other proteins. So can you guys see how this gene regulatory sequence, even though it's not making proteins itself, it's non-coding, it's affecting phenotype, right? Because it affects the transcription and translation of other genes. Very good. I want all of you to make sure when you're talking about non-coding DNA, you mention gene regulatory sequences. That's arguably one of the most important examples of non-coding DNA. Good, we've already gone through the rest, so read it in your own time. But let's move on today. All right, let's go and do. So I wanted all of you guys to do some of these questions. We'll come back to that now in our own break. All right. Now, this is actually a revision dot point. I think they're being nice to you guys in the sense that if you know Mod 5, this dot point's pretty easy. It's just asking you to revisit and explain how fertilization, how meiosis, and how mutation leads to genetic variation. And I'm sure most of you who've done Mod 5 already know this. So starting with Rithania. Rithania, tell us about meiosis. How does meiosis actually result in genetic variation?
Meiosis undergoes crossing over, random segregation, and independent assortment, which leads to genetically varied cells.
Good. Now I'm going to pass on the question to someone else. Crossing over. Mohammed, what's crossing over? What do you understand about that process?
When two homologous pairs of chromosomes, they exchange information.
Very good. Yeah. So it's when the periphery or the adjacent ends of two homologous chromosomes exchange genetic information. All that does is it creates new chromosome combinations or new allele combinations on a chromosome. Because as you understand, on every one of those black arms that you see that I've drawn, there are alleles. When you swap adjacent black arms, you have now created new allele combinations on each chromosome. Very good, right? So crossing over, new allele combinations on chromosomes. Good. Random segregation and independent assortment. I think it's good we're coming back to it because every time I revisit this, it makes a little bit more sense to students. By the time we reach the end of the year, you guys will be masters of this stuff. So going on to Kriti. Kriti, can you explain random segregation to us again?
It's like the random separation of the chromatids, towards different poles in the cell.
Okay, so the random separation of chromatids to different poles of the cell. So you're saying this and this for this chromosome that has two sister chromatids. Is that right or wrong? Ideas? What do we think? Sarah, do you agree with this? The separation of chromatids to opposite ends of the cell?
No, it's actually the random movement of the homologous chromosomes to the opposite ends of the cell.
Very good. Yeah. So very good, Kriti. You're completely right in the sense that we do have chromosomal information going to opposite ends of the cell, but it's not the sister chromatids, right? It's the entire X-shaped chromosome where one chromosome of this homologous pair will move randomly in one direction. I'm just going to close my eyes and I pick this one, which happens to be close to the left one. So this now is going to the left, and this chromosome will now go to the right? Or it could have equally been that they move in the exact opposite direction, where the right chromosome goes to the left chromosome goes to the right. The most important thing is they must randomly segregate, move to opposite ends. Very good. What is independent assortment? Let's go to Matthew.
Who else? I don't really know. I think it was just the idea that they all form. So once they form gametes, there's independent segregation when they form gametes. So there's a random chance they go into one gamete or another.
What's they? Is it a chromosome? Is it the entire
Chromosome.
pair of chromosomes you see here?
The whole chromosome.
Good, good. But remember, this is the thing I want you all to make sure you do as well. Try to explain it in the simplest way you can. If you can explain a concept, this was coined by the famous physicist who was one of the smartest people on this planet, Richard Feynman. If you can explain it to a year 6 student, you truly understand it to a T. So it's a good way to check whether you really understand what's going on. So good job, Matthew. You did get the idea that there is some independent movement of chromosomes. But to very simply put it, independent assortment simply says that the way these two chromosomes assort themselves or segregate to opposite ends is completely independent to another homologous pair, right? So whether chromosome 1 goes to the left or right is completely independent to the movement of chromosome 2 here, because they're in different homologous pairs, right? According to random segregation, if chromosome 1 goes to the left, this other chromosome in the pair must go to the opposite direction. Their movement is actually dependent on one another because they're inside a bubble, they're inside their homologous pair, right? That's what random segregation is. Chromosomes in a homologous pair going to opposite ends. Independent assortment says the random segregation of one pair of homologous chromosomes is completely independent to the random segregation of another homologous pair of chromosomes. What these two chromosomes do is completely independent to what these two chromosomes do. They're in their own world. Kind of like you and your best friend and me and my best friend. What we do is completely independent to one another, but the activity of you and your friend is dependent on each other because you both decide what you want to do. Whereas a completely different pair make a completely independent, different decision. Good. I want you guys to make sure you don't mis-explain these processes, because examiners, these businesses that make your trial papers, they know what students struggle with because they make it every single year, right? And this, what I've just explained to you, is a very core concept that a lot of students don't understand, often because either teachers don't understand it themselves, or they haven't run through it enough times that it visually makes sense. Okay, good. Does anyone have any questions? Because this will arguably be the last time I explain meiosis until your trial exams come up and we do exam questions. Feel free to ask anything you'd like, but do ask it now.
Isn't the independent assortment, isn't that part of the metaphase, right?
Very good question. See, this is where textbooks have different information, right? I've seen textbooks where it says metaphase. I've seen textbooks where it says anaphase. When I was in year 12, I went with anaphase. Now, I'm fine with you guys writing either, but ideally maybe ask your teacher, because this thing you have to understand, from a marking point of view, whatever your teacher says goes. Okay, in the HSC, you're free to say either, but just to give you all one concrete answer, just say metaphase, right? The reason there's a confusion between metaphase and anaphase, everyone, is because in real meiosis, the cell doesn't go, okay, I'm in metaphase now. Okay, I'm in anaphase now. I'm going to do these things. Everything is happening in one motion, kind of like a movie. And it's us who've decided to say X, Y, and Z happens in metaphase, you know, A, B, and C happens in anaphase, right? But they're all just happening together in confluence. So the chromosomes lining up in this way does happen in metaphase, but the actual movement to opposite ends, remember A in anaphase stands for movement to away from each other, right? So say metaphase in your exams. That's the TLDR for your question, okay? But if you do see anaphase, you know why now. Good. Any other questions? Nice. Cool. I'm going to test you all. Rithania, does this model random segregation and does this model independent assortment?
I think it's modeling random segregation.
So, Rithania, it's not modeling independent assortment? Or is it? Be confident in your answer.
It's
Alyssa,
I
help
think
her out. Is this modeling independent assortment?
No.
No, because there's no other homologous pair, right? Independent assortment is explaining the movement of two different homologous pairs. If you don't have two homologous pairs or more, you can't show independent assortment. So, very quick exam test. If you guys weren't able to answer that, you did not understand independent assortment and random segregation. If you were, or you were thinking that it wasn't, then you're getting the ball. Okay, very good, good. This is modeling random segregation because these two chromosomes are jumping to opposite ends. You could model it even better. So if an exam question said, how could an improvement to this model be made? You could say, well, these chromosomes can move in this combination, or it could have been that this chromosome, so they could have also done another lineup here where they show the red chromosome on the left-hand side and the blue chromosome lining up on the right-hand side. And that's showing you that the blue chromosome will now move to the right, the red chromosome will now move to the left, right? And that's really modeling random segregation because it's showing you the blue chromosome has a 50% chance of going to the right and a 50% chance of going to the left. Good. All right. This is a very important part of the beginning of Mod 6. I just want you guys to have a bit of a weighted picture on what Mod 6 is all about. Everything you've learned until now is important base knowledge, but it's not what they tend to ask in the Band 6 or tough exam questions. So does anyone know what's actually, in your exams, for Mod 6, what do they commonly ask? What topics do they actually ask you all that are very high yield? Does anyone remember, is anyone who's looked at the syllabus? It's always good to have a picture of the whole syllabus. What topics do you actually get asked about in Mod 6 in your exams?
Genetic change? Like
Yeah,
how
so
the how introduce into the population new elements.
good, yeah. But I would say there's an even more high yield section than that. So genetic change is what I'm about to explain to you all now, this lesson. Anyone else? What's the end of Mod 6 about? Because I would actually say Mod 6 is kind of like a pair, right? What you learned, you're currently up to here. This stuff is important. It's important to know how to read those mRNA to amino acid tables, right? It's important to understand meiosis diagrams and models. And it's important to know what the mutations and causes and effects are. Those, what I've taught you, out of what I've taught you, those 3 points are the most important. What's the rest of mod 6 about that's so high yield? Okay, since we aren't sure, I'll tell you. So when I mention these words, will perk up in importance, because this stuff, genetic and reproductive tech, and how that affects biodiversity is probably the most important part of Mod 6. Your long response 6-7 mark questions will be about reproductive and genetic technologies. So for now, keep it on the back burner, but as long as you all can appreciate everything we've done so far is important, but not as important as the section we're about to jump into. Good. So let's talk about genetic change, right? Evaluate the effect of mutation, gene flow, genetic drift on the gene pool of populations. Why do we call it a gene pool again, everyone? Anyone tell me? Let's go to Mohammed. Any idea why we call it a gene pool for a population?
It's all the variations of a person, all the different combinations.
Yeah, good. We're getting there, right? So it's all the different variations. And I think what he meant by variations is all the genes and all the variations, which are alleles, right? So the reason it's called a gene pool is because when we have a population, every organism has a set of genes, right? You all have the height, eye color, hair color, protein synthesis genes, but you also carry alleles. And so does every other organism. You all carry 2 alleles for a gene. And the way we think about it is when 2 organisms combine and have an offspring, their alleles flow on to the offspring, where each parent contributes half their alleles. And now you can start to see that from parent to child, alleles are starting to flow on. And when that child has another child, alleles continue to flow on. So it's called a gene pool because every organism has, 2 letters. And as letters just combine and recombine, combine and recombine in every future generation, kind of like a stream or kind of like a pool. It's to show you it's a liquid process. It's a fluid process. Okay. So a gene pool, the exam definition, all you all need to have, refers to all the genes and all the alleles in a population. Okay, now what's high yield about this point is you should all be, from Mod 5, confident in calculating allele frequency. If you're not, go watch the lesson recording where we talk about allele frequency, or go read the textbook or read your notes, because that is very high yield. Most trial papers will either have an MCQ or a quick short response on calculating allele frequency. Okay, because if you know how to calculate allele frequency, you truly understand why it's called a gene pool. Good. So now this question is all about how can this gene pool change? I know it's all listed here, but I need some explanations, right? So when we say the gene pool is changing, we're talking about allele frequencies. That's in reality what we're talking about, right? So I want you all to explain to me how mutation can change allele frequency, how genetic drift can affect allele frequency, etc. So, Matthew, we'll start with you. How does mutation affect a gene pool?
It introduces pretty much new alleles into the gene pool.
Very good, right? So as long as we mention mutation leads to the generation of new alleles, I'm happy with all your answers, right? That's the key thing all of you need to state. Mutation is the only way you can create new alleles. Every other option here is not about new alleles being produced in the population. It's about alleles moving in or out, or alleles just being control alt deleted from the gene pool, right? Which we'll talk about soon. And then the environment selecting alleles that are good and selecting alleles that are bad, which is natural selection, right? Now, Can you hit the raise hand icon? Who's learnt gene flow at school? Good. All right. So I guess it's interesting. We get ahead of school. We only have one class a week. So either the schools, what I've usually found is schools give a lot of worksheets or presentations, or there's not actually a lot of active teaching time versus you just doing work. So I think that's why we're ahead of school. Tracy, have you learnt gene flow? And if you have, do you mind explaining it to the class? What is gene flow?
Our teacher has like a really brief teaching about it, but
Yeah.
I think it's referring to different organism like moving in or moving out because of some kind of reasons.
Yeah, that's literally it. Gene flow, I think it's too fancy of a word. Gene flow is literally talking about immigration or organisms entering the gene pool, or emigration, organisms leaving the gene pool, right? And it makes very logical sense. If organisms enter the gene pool, what if alleles they carry increase in allele frequency? And when that same organism leaves the gene pool, the alleles are removed, and the alleles, or the allele frequency of the allele, decreases. That's all gene flow is, right? It's kind of like showing you that there's water flowing into this pool and out of the pool, but the water is genes, right? That's why it's called gene flow. Gene flow literally means immigration or emigration. When you went on a holiday, your allele frequency in Sydney slightly dropped because you disappeared, right? And when you re-enter Sydney, your allele frequency increased in the population. Whatever alleles you carried, that was actually a perfect example of gene flow. Just a fancy word to explain immigration and emigration. Now, the concept that's a little bit harder to understand is genetic drift. And so for us to actually understand genetic drift, we need to understand natural selection. By this point in year 12, all of you should have a good definition of evolution by natural selection. Let me ask, Sarah, do you want to give us your explanation of natural selection? One letter to give it your best go.
It's the change in species through like development.
Change in species through development. I think the point with that definition is it's vague, right? What are we talking about in terms of development? Is it neurodevelopment? Is it biochemical development? Is it development in a lifetime? I'm developing in my lifetime. I'm getting older, hopefully a bit smarter. So what do we mean by, let's start with Kriti. Give us your definition of natural selection. I'm going to keep going until someone gives us an exam full mark definition.
It's like the process in which organisms like change and adapt to changes, I guess.
Okay. Now, I'm playing devil's advocate here, right? But the change and adapt to pressures, you could apply that in the workplace, right? I have a lot of pressures. There's surgery itself is a pressurous approach. So I think it's again a bit vague, and I wouldn't use the word organisms adapt. I don't like to use that word because yes, we do adapt. We adapt mentally. We sometimes adapt slightly physically, but we don't adapt in our lifetime according to evolution, right? The theory that we evolve and we adapt in our lifetime was what's called Lamarckian evolution, right? That amphibians occurred because fish learned to adapt to get out of water. That's not what happened. We do not adapt in our lifetime. We are born with traits, and they happen to be an adaptation if they're favorable for the environment. I'll give you a perfect example. I have slightly darker skin, right? I was born with it. Going out into Sydney sun The sun is a selective pressure, and having darker skin or melanin is an adaptation. But I didn't get darker skin because I was in the sun, even though it gets slightly darker, it doesn't get significantly enough, right? I was born with the trait, and it became an adaptation because of the environment. You put me in Europe, and I'll get vitamin D deficiency, weak bones, and disease. Suddenly, my trait is no longer an adaptation. So try not to use the word organisms that adapt. Don't like that in your definitions. Tracy, do you want to give it a go?
I think it's the selective pressures, which includes like biotic and abiotic factors, which leads to the selective pressures to a population, and then which would introduce like the organism with the favorable trait is going to be selected in the population. Thus, like yeah, it leads to the population, like the favorable trait continues.
Good. I like how you used a bit more key terms. Selective pressures, favorable trait. I'm going to polish your definitions, okay? Evolution just means change over time. Natural selection is a mechanism to explain evolution. That's what I want you all to understand. Okay, so you need to explain how organisms change over time according to natural selection. Now, the first thing I didn't hear from everyone or anyone was that we need to have a genetically varied population. If you have no variance, if everyone's identical, either everyone will survive or everyone will die over time. You will not get continuity of a species. If everyone dies or everyone survives, that's not really change. The first thing you need to have is genetic variation in the population. The next thing you need to have in this population is selective pressures from the environment, which, as Tracy said, can be abiotic or biotic. You don't need to mention that in your definitions, but as long as you mention the word selective pressure, I'm happy. Okay, the next point needs to be about how the selective pressure will allow organisms with a favorable trait to survive, reproduce, and increase the frequency of their alleles. Whereas organisms with an unfavorable trait will not be able to survive, it will not be able to reproduce, and their traits or alleles are deleted from the population. As you can imagine, that will lead to a change over time. Right? We'll go from a 50% of the favorable and 50% of unfavorable trait to 99% of organisms having the favorable trait, and a very small percentage carrying the unfavorable trait. This leads to a change over time. That is your definition of evolution. So if you have the word genetic variance, selective pressure, and you link that to favorable traits, and finally a change over time, you have explained evolution. Any questions about your definitions? I know you will understand it, but understanding it versus writing it in an exam to get full marks is a different game. Good. So now that we understand natural selection, which is explaining change in a population's allele frequency due to selective pressures, and I want you all to understand genetic drift is kind of the opposite of that. Genetic drift is when you have a change in the allele frequency of a population, not due to selective pressures, but because of random chance. That is the definition of genetic drift. Change in allele frequency due to random chance. Key example of this, one example we can think of, thinking about Australia, has there been any major natural disasters? But New Zealand, right? New Zealand, Christchurch, is at the junction of tectonic plates to the point where there's a lot of earthquakes. Same thing with Japan. When you have a sudden earthquake, everyone, and a lot of people die, are they dying by random chance based on where they were at a point in time and place, or are they dying based on selective pressures? What do we think? Let's go to Matthew. earthquake or a natural disaster, Matthew, does that kill organisms just based on random chance, or does it actually select favorable and unfavorable organisms?
Just random chance.
Random chance, right? And do you all agree that's going to change allofrequency as well? But it wasn't necessarily due to genetic drift. It wasn't due to people entering or leaving the population via migration. And it wasn't because new alleles were being created by a mutation. Hence, it is genetic drift. Genetic drift is change in allofrequency due to random chance. Perfect example of that is a natural disaster. Tsunami, an earthquake, tornado, any natural disaster can just suddenly change allofrequencies. Okay, good. It's all here. Changes in the gene pool due to random chance. Okay. Now, I explained one textbook example of this. So for natural disasters, we use this analogy of the bottleneck effect, right? And what this analogy is showing you is this is the original gene pool of the organism. And all these bottles have, imagine there's a bottle with multiple different colored marbles, which represents the population, right? And the different colors represent the different alleles. Now, when you pour a bottle, the bottles at the balls at the neck of the bottle will pass down, but the balls at the end of the body of the bottle will not, right? And which marbles were at the top of the bottleneck versus which marbles were at the base is just completely random, right? And so it's showing you that the marbles that randomly happen to be at the bottleneck will be able to pass into another container, survive, and completely have a different gene pool compared to the original population. Whereas all the organisms that just due to luck were not at the bottleneck, all those marbles, we remove from the gene pool. Okay, so that's what we mean when we talk about the bottleneck. Okay, the bottleneck analogy is just to show you that the organisms or marbles at the neck of the bottle survive and they lead to a completely changed allofrequency. Good. So the bottleneck effect is used to explain natural disasters. And you'll actually have to say this in your exams, right? So you'd have to say an example of genetic drift is the bottleneck effect when natural disasters change allofrequency. Good. All right. The other example of genetic drift, everyone, so we've spoken about the bottleneck effect. The other example is the founder's effect. And again, this is due to some kind of environmental change. And it's also very simple and easy to understand. Imagine this is a population. Imagine again, there was another huge tectonic plate motion, which led to a huge separation between the two populations, right? Now, let's just imagine, we've got a few blue marbles, we've got a few purple marbles, which represents the other trait. You can clearly understand if you have random separation of the original population, this small subpopulation that now has to have found their own group has a completely different allofrequency to the original population. If you count the number of blue to purple balls, you can see there's 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 7 blue to 1, 2, 3, 4, 5 purple, right? But in this new population, the allofrequency is 3 blue, or 2 blue, sorry, to 1 purple. That is a different allofrequency. And so we call this new segregated population the founding or the founder's population. Hence why this effect of segregation of the population is called the founder's effect. Okay, so key things here, genetic drift, there are two mechanisms: the bottleneck effect, natural disasters, and the founder's effect, where there's geographic isolation or separation of the original population into two groups. Okay, And the key thing I want to hear in your exam answers is these groups can no longer interbreed with one another because of this geographical separation. If you can still migrate to the other population and interbreed, you haven't actually segregated the two populations. Okay, any questions so far? Good. Good. 2 exam questions to see how well we understood this. And just hit the raise hand icon once you have an answer for both questions. I'll ask a few people at random. Good. Get some water. All right. Let's give it a go. So let's get, all of you have been contributing, which is amazing. So I can't really pick anyone. You're making my job hard. Let's just go to Alyssa. Question 18. I'm going to just get each one of you to tell me about every option. So Alyssa, what do you think about option A? With question 18?
I think it might be the answer.
Why? And why do you think it might be an answer?
Because gene flow is like the immigration and emigration of the alleles, I guess, when of the organisms and for their alleles when they're moving in and out of the gene pool. So then some of the alleles would be lost when they like move away from it.
Okay, so I think what Alyssa did well here was she identified, okay, there's 2 gene pools. I want you guys to always draw diagrams so you understand this, right? So if I was a student, if I was doing this question, I'd be like, okay, gene flow is occurring between 2 genetically different populations. So maybe I would just write population A and B to show that they have different alleles, okay? But it's of the same species. Why does that matter? Of the same species. Sarah, what does it mean when you're the same species? What can you do?
Same
definition would be-
species.
Yeah.
It would mean like they're genetically similar, so they can like they can change. I mean, they can reproduce together.
A donkey and a horse can reproduce, but they're not the same species. So what I want you all to understand, definition of a species is clear cut. There's no fluff around it. This is why I like biology. Unlike English, where you can interpret one thing in 50 different ways, and there's no real answer to which one is better than the other. Science has very clear-cut definitions. A species is a group of organisms that can reproduce to make fertile offspring. So all this point is telling you is that whichever organisms jump to the other population will interbreed, make new offsprings, and change allele frequency. Is everyone with me? Right? If you had two different populations of different species going across, to each gene pool, it's not going to affect the gene pools because they won't be able to reproduce. And that would be a pretty good exam question to test whether you understood the definition of a species. All right, you need to understand organisms are moving across one another, right? This is what is happening. Alleles will be lost. Now, this is completely right. When organisms move out of a population, alleles will be lost. But Alyssa, will they be lost due to genetic drift or due to gene flow?
Due to gene flow.
Good. So that's why this option is not right. Does that make sense? So we need to be very critical of every single option. Next, going to Rathania. B.
I don't think B is correct.
Good. We clearly said A and B have different allele frequencies. So when organisms of B move to A, then a further change the allele frequency, right? So the point that they're not going to change is silly. The very dot point says gene flow is an example of how you change gene pools or allele frequencies. Next, the gene pools of the populations will not change. Of course, again, allele frequencies and gene pools are almost synonymous, right? The only difference, everyone, is a gene pool refers to all the genes and their allele frequencies. So you can kind of think of it like this. The umbrella term is gene pool. There is gene 1, gene 2, gene 3, and gene 4. And every gene has a frequency of allele 1 or allele 2 for the HSC. In real life, there's so many alleles for one gene. But can everyone appreciate the difference between allele frequencies, which is here, and the gene pool, which is this whole thing? Good. When you change allele frequencies, you change gene pool. So B and C are wrong. So even without thinking, I don't even have to read option D. You of course should, but I know D is the right answer. And does it make sense? Yes. Because when two organisms are migrating, just think about Australia, perfect example, right? Australia is very multicultural because there are all these different ethnicities which migrate in and also migrate out. And the end thing that happens is Australia ends up looking a lot like the broader international world, right? There are subsets of people of every single ethnicity to a similar percentage, right? Whereas Australia in the 1700s or 1800s was more Anglo-Saxon or Caucasian predominant. Good. D is the answer. Good. Let's try doing 6 a bit faster. Let's go to Trissy. Mohammed, I'll get you to start with A. Mohammed, what do you think about 6A?
I think if individual leaving the population equals to the number of individual entering, it's not going to remain constant because the two different alleles of the individual leaving and the individual entering is different. So if they go into a gene pool, it's still going to change the alleles.
Good explanation, right? Which is, again, I always draw this. It's very easy. Gene pool is the circle. if the number of organisms leaving is equal to the number of organisms entering, the allele frequency must be the same. But that doesn't make sense, because what if all the organisms leaving have the capital A allele, all the organisms entering have the little a allele, which is exactly what Shruti said, different alleles, right? What that's going to do is increase the frequency of the little a allele. So again, A is not right. Do you see how we're using thought experiments? Really helps with bio, really helps with chem, helps with physics, okay? Arguably the most in physics. Einstein changed the entire world just using a thought experiment. Does anyone know the special relativity thought experiment, just for interest's sake? No? Well, maybe I'll ask it to you in the break. But all right, let's move to option B. Matthew, what do you think about option B?
That's correct.
Why do you say that?
Because you have a greater number. It's like saying you have a smaller population, so even though the frequency could be the same, you have a larger number of organisms with that allele. So therefore, if you, let's say you cut the same section of a larger population or a smaller population, the larger population won't be as affected.
Good. Yeah, I'd say, yeah, it's quite a good thought, right? So, like, for example, if we deal with a population of Sydney and we split it in half, you still more or less have a very similar allele frequency. Well, it really depends on how you cut Sydney, because Sydney is a very interesting example where there's little hot pots of different ethnicities in different parts of Sydney. But assuming you just cut it half, what will end up happening is more or less you'll have a similar ethnicity or allele frequency. Whereas if you have your classroom, for example, and there was genetic drift or natural disaster or the founder's effect, and only 5 people survived, for example, there's a decent chance it could all be the exact same ethnicity. And that would completely change the allele frequency, right? And this is just to show you, this is why thought experiments help. With a large population, splitting in half, you more or less will have a similar frequency. Tiny population, if you split that in half, there's a very good chance the 4 or 5 organisms that survive could all have the same trait, which would completely change the allele frequency. Whereas it's very unlikely that a natural disaster will wipe half the population of Sydney out and everyone alive would have the exact same trait, height, hair color, etc. Good. Very good. So I'd say B looks good, but remember, you have to go through all the options and eliminate everything else. So what do we think, Kriti, about C?
Maybe because when like there's a natural disaster, it'll like wipe out, like for the bottleneck, it'd wipe out the ones which won't be able to survive, and it'll keep the alleles which can reproduce, and they'd have the allele to
Good thought.
fight against.
Good thought. Let's use a natural disaster as an example, right? Let's say right now there is a huge tsunami that just wipes out half of Sydney. Do you really think, Kriti, the organisms or the people that survive would be better equipped to survive against future tsunamis? Or do you think just by chance the people that don't live on the coast would survive and they actually don't have a survival advantage against future tsunamis?
Oh, they won't have a better.
Yeah, I think it's more the latter. It's just random chance, right? Time and place. If I happen to be at the harbour, I would most likely pass away. Whereas if you went on a holiday down to Northern Territory that day, then there's no chance that you're going to be harmed, right? So the key thing I want you guys to understand is bottleneck effect is change or natural disaster that changes allele frequency. It has nothing to do with natural selection, which is the process that allows organisms to be better equipped to changes in the environment. Right? Natural selection is what allows that to happen. It selects organisms with favorable trait who will now be able to survive better against that selective pressure in the environment. So C is not an answer. D. Final one. Let's go to Mohammed. What would you pick for option D? What do you think about this?
I think it could be right, but at the same time, it's a bit confusing because it's if a individual organism develops a mutation that might or might not be better for the environment, it might not carry on.
Good, good. I like that answer. Mutations can be good or mutations can be bad. So that's one point here. But Mohad, what do you think about the part that I bolded there, which is that organisms develop mutations in order to better suit themselves against the environment? Is that how mutations arise? So if you expose me to a selective pressure, will I just mutate to better suit that environment?
No.
No. Very good, right? Mutations happen due to random chance, right? They happen due to errors in my DNA replication process. And they can occur due to radiation from sun exposure, or they can happen due to ingestion of charcoal chicken, which apparently increases your risk of colon cancer very slightly, not as much as cancer. Cancer is really bad. I've seen so many patients get surgeries that I wouldn't wish on my worst enemy because of smoking. Some surgeries where you have to literally cut the neck, break the ribs, cut half the chest, and cut the chest here as well to take out all the esophagus for esophageal cancers. And those patients literally can't even eat proper food for the rest of their lives. It's crazy. But as long as you all understand, mutations, random. Okay, it's not in order to better suit yourself for an environment. So D is completely wrong. Good. The answer is B. All right, you guys can do the rest of these questions, open questions in your own time. A very, very important question to do is question 3, the 4-mark question. I don't know why it's come as a dot point. Just be analyze how the mating program would impact gene flow and genetic drift. We won't do it this lesson, and even question 4. This is how your exam questions will be. Can you all see how it's showing you frequency of alleles? It's asking you to graph the data. It's asking you to justify why those alleles are changing. That's kind of exam questions you'll all get in your trial exams. Okay, good. All right, we've hit the hour mark. Make sure you all stand up, walk around. I have to keep looking at a screen. Get some water, and then we'll recap at about 3:15, 3:16. Okay, good. Make sure you all take a break, because otherwise looking at a screen and not talking can get very tiring. Good job. See you guys all in about 5 minutes. Okay, when we're back, can we hit the raise hand icon? Good. All right, I think Alyssa's sent a question in. So let me share my screen and we'll run through it. And this is also what the break in the beginning of class is for, guys. Even though I quiz you on the beginning of class to make sure you understand things content-wise, I think the way, and I've said this multiple times to those who've been here a while, the best way you can make the use of classes is do exam questions every week. And whatever exam question is hard for you to mark or you don't understand, send it in the Teams channel before or as soon as class starts. Make sure you have the screenshot ready, and then we can go through it together and I can show you how to think and break down the exam question. You guys can watch a bunch of YouTube videos and learn biology. That's not the main thing that we're all here for. It's how do you then apply that for the HSC exam and the questions and how to think to get full marks. That is the challenge of, going from a high band 5, a low band 6 student doing really well in a subject, right? Good. So Alyssa has posted a question from Charles. I think this is the last for Charles. And let me just screenshot it so we can go through it together. Good. So what I'm going to do is get all of you to give this question a go and hit the raise hand icon once you've got an answer. Maybe in the interest of time, I'll just start breaking it down with you guys, right? So let's read it together. Group of biologists at Taronga Zoo want to mate their endangered penguin. I would literally at this point draw a little penguin. Okay, with a penguin from another zoo. So this penguin is, I guess it's gene pool, or maybe it's from a gene pool of penguin in Taronga Zoo. I hope they don't only have one penguin, that'd be sad. With another penguin from another zoo, so another gene pool, to assist in the conservation of species. And this is an endangered penguin, so they'll put E. They've sent DNA samples from 3 possible mates. The results of an electrophoresis gel in which the DNA of Taronga Zoo's penguin, lane 1, I'm going to call this penguin E because it's endangered, was compared with 3 possible mates. So they must be from the other zoo. So these are all from zoo 2, 3 possible mates. Okay. Now, first of all, how do we interpret this? What are you being shown on an electrophoresis gel? Let's go to Sarah. Sarah, what are you being shown in this image here?
The gene of the organism.
Yeah, it could be a gene that's been cut up by restriction enzymes to produce these fragments, right? The key thing you all need to understand is PCR was a reaction that was done before this. It amplifies the DNA. We're looking at the same section of DNA from these 4 organisms. You can almost think of each band as a gene or like as an allele, right? If you're trying to mate 2 organisms to increase genetic diversity or maintain genetic diversity, you want them to be as different from one another as possible. So you want to pick the organism which has the least bands lining up with organism 1. And Matthew, which organism would that be?
I would say 2, but
Oh, I'd say 2 has almost every band lining up with organism 1. Right? It's got 2 bands lining up with organism 1. So actually quite genetically similar. And I'd say a similar answer for organism 4 and 1. Both got 2 bands lining up as well. But when you look at organism 1 and 3, I can appreciate the only band that lines up is probably just this one here. Every other band between organism 1 and 3 is different. So they have the least bands lining up. They're thus the most genetically different. I would pick organism 3 to mate with organism 1. Good. How will the cross in question 13 affect allele frequency in the population? So you pose this question. What's your rationale for what you picked?
I thought it would be B because there would be more alleles in the population as a result of the cross. But I think they were going for something else.
No,
Yeah.
I completely agree with the statement you said. This is why we have to be so careful. This is the thing with biology, right? Sometimes the statement that's made is completely right, but it doesn't explain the question being asked, and that's why it's wrong. And sometimes your justification for what you picked is completely right, but it doesn't actually answer the question, right? So for Alyssa, her point was, yeah, well, of course, I'm getting a penguin from another zoo, and via gene flow, I'm entering it in and causing it to mate with this penguin. I'm increasing the number of different alleles. But this question is not asking about the number of different alleles, it's asking about allele frequency in the population. And if you had to guess, when they're talking about the population, they're probably talking about the endangered population, right? Now, logically think about this, everyone. In this endangered population, they haven't told us there are any other penguins. They can almost assume this one penguin is the endangered species, right? So all of its alleles represent 100% of the gene pool. But now, when I add another penguin which has completely different alleles, right, the frequency of this endangered penguin's alleles will drop from 100% down to making up 50% of the gene pool, right? Because there's other new alleles, such as this allele introduced by that same penguin. So technically, I would say, if you think really hard about this, the answer to this is there'll be a decrease in allele frequency. There'll be a greater number of alleles, but that's going to decrease the frequency of each allele. Instead of it being 100% of the gene pool for that one allele. Remember, having high allele frequency is not good. If you have a very high allele frequency, most organisms have that allele, and there's not much genetic diversity. Whereas if you have more of a 50-50 split, or you have multiple different alleles, you have 4, 5, 6 different alleles, each allele's frequency will be very low, but there'll be a significant genetic diversity in the population. And so learning point here is that a high allele frequency does not mean a high genetic diversity. In fact, having a low allele frequency because of multiple alleles means high genetic diversity. So this will actually decrease allele frequency of those endangered alleles that penguin 1 has, because we're introducing penguin 3's alleles in. Alyssa, does that make sense?
Yep.
Good. Does that make sense to everyone? Good. All right. That was a good question. Thank you for sharing that. See how they tested both mod 5 electrophoresis and PCR with mod 6 allele frequencies as well. That's what your exam will look like. See, all this stuff, it's all biology, it's all combined. There's so many cool questions to ask you guys, which will mix everything up together. And in a 3-hour exam, that's what they're going to do. Tracy, question?
I get a bit confused between the difference of new alleles and allele frequency, because I remember mutation is the only way to introduce new alleles into a population. But then just now we were talking about allele frequency, and then I kind of mixed up what's the difference between higher allele frequency and a new allele, or what's the definition of a new allele? Does that make sense?
Right, that makes a lot of sense. So your question is quite literally, well, if we have this population, this organism moves in with new alleles, or alleles that aren't present in this original population. If this organism comes in with alleles that aren't present in this original population, it's technically introducing new alleles, right? And you're right, it is. But wherever this organism came from, whatever gene pool it came from, would have already had those alleles. It would have got it from its mother or father. It would most likely still be in that population. Would have got from other organisms. So that allele would not actually be new from scratch. Mutation is different because you'll have a population And without migration, there will be a new allele that appears. Where did it come from? Nowhere. It was just generated as a new allele, and that's what mutation does. So technically, yes, gene flow can introduce new alleles to a population if that population has never seen that allele before, and if that organism came from a population that had that different allele, right? But in mutation is the only way to generate new alleles from scratch. So in your exams, try to only state mutation is a mechanism to create new alleles. Don't talk about gene flow creating new alleles, but you can talk about gene flow introducing alleles that are not familiar with it or are familiar in the new population. Does that make sense?
Yeah, thank you.
Good, good. So TLDR, mutation is the only way to make new alleles. Technically, gene flow can introduce alleles that are new, but they're not actually new. They come from another population. Good. All right. Mutation inquiry question 2. How does mutation introduce new alleles into a... There we go, right? We've technically already answered this inquiry question, not just then, but also when I taught you about how mutation arises. But do you guys all see how this is an inquiry question? It's to show you how important this statement is. Mutation is the only way we can introduce or generate new alleles in a population. Okay, good. Well, someone answer this question for me. That's what the inquiry questions are for. So let's go to Mohammed. Mohammed, how does mutation introduce new alleles into a population?
From mistakes that occurred randomly that create new alleles.
Good. So you've said that mutations can occur randomly. So spontaneous mutations are what we're talking about there, which occur from errors in DNA synthesis of mitosis. DNA synthesis of mitosis. Okay, sure. How does mutation introduce new alleles into a population? Yes, through spontaneous errors during DNA synthesis. But how else do mutations arise, everyone? You all learned this as well. Quite a simple question. Let's go to Kriti. How else can mutations arise, Kriti?
The change in the base sequence of DNA.
Yeah, that's what a mutation is. But remember, we're talking about causes. What causes that?
Or about the environment.
Good. Yeah. So Mohammed, tell us a bit more then. What from the environment can cause a mutation?
Newer environments could change the DNA, which could occur as mutation.
Yeah, good. But we're still being vague here. So I'm going to pass it on to Sarah. So what parts of the environment, what does a syllabus break it down into as the 3 causes that can induce a mutation?
So there's biological, physical, and chemical.
Good. That's what I want to hear. Categories. Remember I told you, once you have categories, it makes sense. Then you go, okay, physical, what do I mean by that? Ionizing electromagnetic radiation, right? X-rays, radio waves. Sorry, not radio waves. Radio waves are very low frequencies. X-rays, UV, and gamma are the only 3 waves in the entire electromagnetic spectrum, which includes visible light, infrared radiation, which is literally just heat. When you feel heat, you're absorbing infrared radiation. That's what heat is. Microwaves, radio waves, those are the only 3 types of waves that cause mutation. Chemical, we spoke about smoking. There's also asbestos. There's also nitrosamines in smoked foods, which is why I keep bringing up charcoal chicken. Mind you, I eat a lot of charcoal chicken, so it's all about moderation. When you do medicine, you realize everything is bad for you in some way or form. And some bad things are technically good for you in very, very rare, unique conditions. For example, smoking has been shown to be protective in a form of inflammatory bowel disease called ulcerative colitis. But of course, we don't tell people with that condition to smoke because it increases their risk of cancer as well. So it's all just, everything is bad for you. You learn once you read up enough, right? But everything to a different degree. Biological, viruses like HIV, okay? HPV, which causes cervical cancer. We also have transposons, which are jumping genes that are usually non-coding DNA, but can jump into coding DNA and thus change it. So then the next thing we need to answer is, well, yes, all these things cause mutation, but how does a mutation cause new alleles? It's a very simple point, but you all need to say mutation changes the DNA sequence, which leads to new alleles, and then you can link that to change in phenotype in a population. Okay, good. So we've answered this inquiry question. Does anyone have any questions about how mutation introduces alleles into a population? Good, good. All right. Let me just pause and have a look, because this should technically be inquiry question 3. Yeah, see, this is inquiry question 1. It should not be repeated. I think that's the error in the notes, because it actually doesn't answer. This inquiry question should be biotechnology, everyone. Let me go back. This should be biotechnology. Let me quickly pull up what the actual inquiry question is, but this is not the.
I think it's how do genetic techniques affect Earth's biodiversity?
Oh, great. Is that in your book, Tracy?
I'm not sure, but it just says inquiry question 2.
Oh, so this is not what it says on your page in the notes? You have.
I think it is, but I think the inquiry question is just a bit different.
Yeah, because this is the inquiry question 1. Yeah, so key thing, coming back down here, this inquiry question should be biodiversity and biotechnology. So I want all of you to just note that down, if this is what you're seeing on your page as well. I might have downloaded an old copy of the notes. But yeah, good. Key point is this inquiry question is about biotech and biodiversity. So let's define the two. So biotechnology is an extremely vague term. I want you all to understand that. It's not just the use of living organisms in tech, which is what it sounds like. And that's because the word technology is vague. What is a technology, right? Biotechnology is the use of any living organism or its byproduct. So it doesn't even have to be the living organism itself. Any product that's made by that living organism as well, in any application in health, society, and industry, which is literally every single sector in humanity, right? So if we use living organisms for any human benefit, that is regarded as a biotechnology. I'll give you a perfect example of that. Beer or alcohol production is a biotechnology. The reason is, if anyone's gone on a wine tour, or if anyone's seen, I remember when I was in 3rd grade or 6th grade, we saw how I think convicts made wine. We went on a little excursion and we'd have to step on grapes, and it's the yeast in the grapes, which are on the feet, that will then ferment the sugar in the grapes to make alcohol. And since we're using this fungus yeast and its enzyme called zymase, that's what's present in the yeast, to convert sugars in grapes into alcohol, that is a biotechnology. Stepping on grapes is a biotechnology, as weird as it sounds. That's because we're using a living organism and its byproduct for a use in industry, which is the alcohol industry. Right? So that's a good example of an ancient biotech. Now, for this dot point, as you can see, you need to know examples of past, present, and future biotechs. For your exams, I would only have one example of a past biotech, but have a couple of examples of a future and present biotechnology. Okay, so for past example of biotech, remember this. Sugar can be broken down into carbon dioxide and alcohol. right? So it should be C6H12O6. That should not be the exact reaction, so it should be C2H5OH or CO2. Do you need to remember this? No, not at all. It's just for those of you doing chemistry, you could kind of appreciate this. Those of you who don't do chemistry, as long as you can understand this is sugar, and yeast will act on the sugar or the glucose to make ethanol, which is the main constituent of alcohols, and it also makes carbon dioxide. Now, I'll tell you why the carbon dioxide is important as well. But this is probably the one reaction that if you know, you can explain past examples of biotech. Yeast, which is a fungus, break down sugar into alcohol and carbon dioxide. Now, we use the ethanol in the alcohol industry. Does anyone know where we use the carbon dioxide? The answer is if we expose wheat. So wheat are just plants, and plants contain sugar, right? But the reason we use wheat in this example is carbohydrates are just long chains of sugar. So bread is literally a sugar, but it's just long chains of sugar, hence why it doesn't taste as sweet, but it's still regarded as a carbohydrate. Okay. So what happens in this example is yeast can act on wheat and produce carbon dioxide that produces bubbles that allows the wheat or the dough to raise into bread. Next time you make a sandwich, have a look at the bread and you'll see little holes all over the surface of it. I'm sure you can all remember the last time you ate a sandwich, how there's little holes everywhere, right? Those holes are due to the carbon dioxide that's made in the dough due to the action of yeast on the sugars. Any questions about ancient biotech? There's not much else to mention, except it would be good to appreciate that the indigenous people of Australia, the First Nations people, also used aquaculture as an ancient biotechnology. So that involves creating fish traps made out of plants to catch fish in canals, right? And since they're using plants to create, these meshes and these nets, plants are living organisms. They're being used in some application to help society or people, technically a biotechnology. Okay, good. So I just know about this in case you get an MCQ question on indigenous aquaculture. Good. Any questions? All right. Here is my open-ended question to you. I'm going to skip all of this and come here. What are some modern biotechnologies you know? I'm going to give you all about 10, 15 seconds to think about this. This is such an open-ended question, and hopefully you'll give me some cool examples. I'll be right back. Okay, just going across. Hopefully I get some interesting examples. Atul, I want you to type down an example of a cool modern biotech. Sarah, what's an example of modern biotech?
Antibiotics.
Cool. Why? Why is that a biotech? How do antibiotics use living organisms?
So it kills bacteria by commonly inhibiting amino acid synthesis from bacterial cell wall.
Very good. So you've just told me it kills bacteria and give you the mechanism. Remember, for it to be a biotech, it needs to come from a living organism or it needs to be a living organism. Do antibiotics come from living organisms, and which one if so? Anyone? So open-ended question, not just for Sarah. Where do antibiotics come from? Anyone give me a name of an antibiotic? Especially those of you interested in medicine. Penicillin. I have never prescribed penicillin in my life because every bacteria nowadays is resistant to penicillin. The only time we give penicillin is in pneumonia or lung infections, which are severe. In that case, we prescribe a drug called benzopenicillin. But anyway, a bit about penicillin. It was discovered when a doctor at a hospital was studying how a bacteria was getting its growth inhibited by a fungus. And the name of this fungus that's growing on the right-hand side here was the Penicillium fungus. I thought it was pretty cool. How come every single bacteria that's growing on this petri dish is crapping its pants when it gets close to the fungus? And so it took him a while to purify the chemical made by the fungus. Lo and behold, we discovered penicillin. And this will help you understand why antibiotics aren't the best for you, because nowadays antibiotics either come from other fungi or they come from other bacteria, and they're actually toxins themselves that are inhibiting the growth of other bacteria. And that's why, before prescribing antibiotics, it's so important to know, does a patient get a rash? Do they get nausea, vomiting? Do they get diarrhea with that antibiotic? Right? And that's because antibiotics are technically toxins from other bacteria or fungi. Right? And some people get anaphylaxis from antibiotics as well. So it's very careful what you prescribe. Nowadays, amoxicillin is the main antibiotic most patients get when they're sick at hospital. Good. All right. So we've got antibiotics. Why are they a biotechnology? Because they're produced by fungi or other bacteria. And the chemicals that usually, I really liked your explanation, is they inhibit bacterial growth. And that's usually by inhibiting cell wall synthesis. Now, interesting point, everyone. The reason that most antibiotics do this is because the antibiotics that we've made that have other mechanisms of action, such as impairing protein synthesis or impairing the cytoplasm of the cell, cause significant side effects to people, because guess what? We also make proteins. We also have a cell wall. So making a toxin that impairs those things causes much more side effects. Whereas we don't have a cell wall like bacteria. And so when we make a toxin or collect a toxin from bacteria that kills cell walls, we don't get much of a side effect. Make sense? Good. All right. I think antibiotics are really cool. They've probably saved billions of people by now. Next person, Alyssa, give me a cool biotech you've learned or heard of at school or researched or saw on the internet or Facebook.
Transgenics.
Good, good. So under modern biotechnology, what we can also put as a category is recombinant DNA technology. Recombinant DNA technology. That's technically a branch of biotechnology. So maybe I'll get you to rewrite this. Under modern biotechnology, future biotechnologies, you can put genetic tech. An example of genetic technology is recombinant DNA technology. Recombinant means recombine. We can recombine DNA nowadays from one species to another species. So when we take a gene from one species and we place it in another species, that's a transgenic organism, which is what Alyssa is talking about. Nowadays, we have the ability to get genes from one plant, put it in another plant or another organism. Has anyone had, what is it called? Cotton candy grapes? At Woolworths? No, they're very sweet. Yes, they're very sweet grapes. Also, unfortunately, very expensive. And they're an example of a transgenic species. We get sugar-producing genes from other organisms, other plants, and we place it in the grape. And you lead to a genetically modified grape that has extra sugar and has that vague taste of cotton candy as well. Much more expensive. Why? Because it's using genetic technology. Good. Someone give me another cool example. Matthew, your turn.
Golden rice.
Good. I think golden rice is really cool, but it's cool in a distant way because none of you would think it's too cool. It's much cooler in what it does for developing nations. So golden rice is just rice, but it's an example of a transgenic technology. We took a gene from a bacteria, we took a gene from a daffodil plant, put it in the rice, so it starts to make beta carotene. And that's a precursor to vitamin A. And so this is pretty much rice that's fortified in vitamin A, and that's used in developing countries to prevent vitamin A-related vision problems, right? So that's rice genetically modified to help people in developing countries who have vitamin A deficiency, etc. Good. Good, Tanya? We're missing some really cool examples. What else do we have?
Would insulin be one?
The way insulin is made would be one. That's an example of a transgenic technology. We insert the human insulin gene in bacteria. Bacteria start making insulin. We collect it, purify it, and pharmaceutical industries sell it. And it's interesting because when insulin was first discovered, it was about, is either 7 cents. When it was first made, it was very cheap, and then it became $7, and now it's $174 for a vial of insulin because of big pharmaceutical industries, right? It's just they can up their cost however they want, which is the sad part. Good. Again, it's an example of recombinant DNA technology where we're recombining DNA. What else do we have, guys? Trissy, what else do we have that we haven't spoken about?
Maybe vaccinations.
Very good, right? A lot of vaccines are actually dead or weakened versions of pathogens like viruses and bacteria. And I would probably say that vaccines are probably the single biggest invention in mankind that have saved the most lives ever, which is very interesting. So anyway, so we've got vaccines as well. Vaccines which use, dead or weakened pathogens, and that's to the right, dead, weakened pathogens, right? And that's a very interesting example, which we'll talk about soon. Good. One thing we're missing is reproductive organisms. Reproductive organisms. Okay, and reproductive tech. So for reproductive tech, how about we just go here? Table here. Yeah. Before we go through reproductive tech, I want you guys to answer these two checkpointy questions, and then we'll talk about the different types of reproductive technologies. Okay? Good. I'm going to get some water. I'll be right back. Okay, so question 2 and 11. What's our answer for question 2? Lasagna?
D.
Good. Very easy question. Question 11. What's our answer? Let's go to Matthew. What do you think about 11?
I put A.
Okay. Why did you put A?
Was the idea of artificial insemination is the idea that we can get one cow, take its gametes, and let's say transfer across the world to one from one global area. It's pretty much gene flow.
Yeah, good.
And we're introducing into another cow that's never really had those alleles.
Yeah, okay, good. So we can draw that down. There'll be geographical barriers, and we've overcome those geographical barriers by transferring one gamete into multiple organisms. Yeah, I would say initially when you first do it, you do increase genetic diversity because that bull is from a completely different population. But over time, if you repeatedly do it to the same organisms and you keep using that same original parent one, you would decrease genetic diversity because the same alleles from that male organism are repetitively being reused in the second population. So I'd say the answer about whether it increases genetic diversity is dependent on time, right? So here I would say theory, it could short term, but long term it wouldn't. And this question is a bit vague in telling us how many cows it's been transferred to, because that will also affect it. If we transfer it to every single female cow, eventually it would decrease genetic diversity as well. But it's a yes, could be an answer, but there is a small flaw in that it would increase it only in the short term. What about grafting two or more fruit trees together? And just to mention here, everyone, this is an example of a reproductive technology, which is again an example of a biotechnology. Okay, grafting two or more fruit trees together. Does anyone know what technology this is? Grafting two plants together. What is that?
Artificial vegetation?
Yeah, artificial vegetation itself is not a commonly used term. This is an example of plant cloning, right? So what you can do is if you want to clone a plant, you take a piece of its shoot system off. This is plant one. If I want to clone plant one, a very easy way to do it that agriculturalists use is they take a bit of that shoot system of plant one and they'll graft it on. Grafting is just when you make a really clean cut in the bottom half of one plant, another clean cut in the top half of the other plant. That is kind of the inverse of complementary. And then you bandage them together and they actually start forming connections with one another. And you technically form a clone. Okay, this new plant too, even though it has the base and the roots of a completely different plant, it still has the same shoot system and it can be regarded as a clone. So this is called cutting and grafting. And this is an example of cloning plants. And cloning is a reproductive technology. Okay, so one thing to know about cloning is you're making a copy of a plant. I wouldn't say it's increasing genetic diversity. So I put a cross here because this is plant cloning. Yes? Cross. What about C? Artificially pollinating an endangered plant species to increase seed production. What do you think about this option? Let's go to Tracy. What do you think about C?
I think not right because it's still the same, like it's still the same seed, which won't really introduce like genetic variation.
Yeah, you're right to an extent, right? So we need to understand every seed, a seed is a zygote, Everyone with me? And zygotes are made from two gametes, and gametes are made from meiosis. So every single gamete from an organism is different. So every single zygote or seed from a plant is also different. A seed of a plant is just an offspring, and every offspring that two parents make will also be different. In the same way, every offspring that a plant makes will be different. So they will be genetically varied, but this question is not about genetic variance, it's about genetic diversity. And technically what we're still doing here is that causing this endangered plant to just make more seeds or more organisms which contain its alleles. So again, I would say this is an increasing genetic diversity. It's preserving genetic diversity by trying to keep endangered organisms alive. But you're technically just producing new organisms with those original alleles, right? So you're just persisting those alleles in the population when they're about to disappear in the endangered organism. So I think this is more preserving genetic diversity rather than increasing it directly. What about option D? Producing a transgenic organism. Very short sentence, straight to the point. Alyssa, what do you think about D?
I think it also could promote genetic diversity because you're taking a gene from one organism into another, and then when that transgenic organism like reproduces, I think its offspring could also have that gene.
Guys, what is the definition of genetic diversity? Let's go to Sarah. How would you define it?
So genetic diversity is where there's various diversity of gene and allele combinations.
Good, in a species. That's what I'm here. Genetic diversity is a number of genes in a species, right? So when you produce a transgenic organism, you're literally taking a gene from one species, putting it in another species. That is the clearest way you can increase genetic diversity. So I would say A is kind of right, but the fact that they've said from one bull is a bit scary, because when you just use one bull and you inseminate multiple cows, you can actually decrease genetic diversity because the same one favorable set of alleles is being used, right? It's going to increase the frequency of this bull's alleles in the population, but that may not increase genetic diversity if that's the only allele in the population. Whereas D is the cleanest, clearest example of increasing genetic diversity. You're cutting a gene from one species, putting it into another who's never seen that gene before. You're increasing genetic diversity. So your answer is D. This is just to show you, most MCQs, there'll be two answers that are pretty sure you're wrong. There'll be two options that you're not sure about. This is where you have to really think, what is the question asking? What is the definition of the terms in the question? And which answers it best? D does. And if you're unsure, try verbally, try saying it to yourself. I often find that when I'm thinking in my head, I come to conclusions that might not make too much sense. But when I'm saying it out loud, right, it forces you to correct or rectify issues that might not make sense. So D is the answer. Good. So next week, I want you all to make sure you've had a read about biotechnologies, vaccination, antibiotics, reproductive tech. The goal is, if you guys are fast, we can probably finish Mod 6. If you guys have read up on reproductive technology, such as cloning, artificial insemination, you can probably finish Mod 6 in about 2 to 3 lessons. So on pace. Okay. Does anyone have any questions? Tracy, question?
This may not be directly related to the one we're doing now, but may I ask if for mutations, like for the questions for mutations, do I have to link it back to like how like natural selection or like how the favorable trait like
It depends. It really depends on the question. If the question is about mutation and continuity of a species, you do need to link it back to evolution. If the question is just about the causes and effects of mutation, if it's about the cause of mutation, you don't have to link it to evolution. If it's about the effects of mutation, well, the effects on continuity of a species is one effect, right? So I think you should link it. But if you're just explaining nonsense, missense, chromosomal mutations and how they work, I wouldn't always link it to evolution. No.
Yeah, okay, thank you.
Good, good. All right, well, I'll see you all next week, everyone. And those of you doing chemistry, just remember we have a 15-minute break, so you can go, stretch, get some water, eat a snack, and then start. Otherwise, I'll see you all next week. Have a good week.
Sorry.
Yeah.
Can I also ask if because I don't think I have the access to the EDX website yet.
Oh, I think Chrissy, I gave you or approved your access. Double check now with Gmail. Check now, yeah, because I did that the other day. Let me know
do I
by
use the personal email or?
Yes, personal.
Okay,
Yeah,
thank you.
Good. All right. And Mohammed, could you stay back? And everyone else, I'll see you all next week. Okay. Bye. Yes. Hey, Mohammed. So let me just stop the recording first. How did you